Hello, I'm Dr. Josephine Yusun Sunga, and I'm head of the Nestle Nutrition Institute. Welcome to the NNI96 workshop on strategies in neonatal care to promote optimized growth and development, focus on the low birth weight infants. From the last session, we learned about donor banking, human milk fortification, and the nutrition for the proper growth of preterm infants. I would like to thank Professor Ferdinand Hashke and our speakers. The third session is on the role of the HMOs and the microbiome in the health of very low birth weight infants, and this will be chaired by Professor Lars Bode. Lars is a professor of pediatrics and is the chair of the Collaborative Human Milk Research and director of the Larsen Rosenquist Foundation and the Mother Milk Infant Center of Research Excellence of the University of California, San Diego. He is dedicated to research on breastfeeding and human milk in general, with particular interest on human milk oligosaccharides. He is the recipient of numerous international awards and has published over 120 peer-reviewed articles. Welcome, Professor Lars Bode.
Hello, and thank you so much for inviting me to not only speak in this session, but also to chair it. It's going to be a very interesting session. Session 3 is going to be all about human milk oligosaccharides, short HMOs, and the microbiome in the context of health of very low birth weight infants. And you'll see we'll introduce oligosaccharides first, then we'll introduce the microbiome, and then in the second part of the session, we'll give you some clinical studies and data on clinical studies, first in the term infant, and then later in the preterm infant. And then we close the session with expanding beyond human milk oligosaccharides, beyond the microbiome, and talk about other human milk bioactives and what the future of this field will look like. So let's get started with the first presentation on human milk oligosaccharides. So what are human milk oligosaccharides and what do they actually do? That's going to be the focus of this first talk. To start off the presentation, let's really take this statement here from the 2016 Lancet Breastfeeding Series, where it says that the death of 823,000 children and 20,000 mothers each year could be averted through universal breastfeeding, along with economic savings of $300 billion US. And really, we are asking the question, why is that? What is in human milk that saves lives? And how does this all work? To understand that and to tackle those questions, we have to understand that human milk does not stand in isolation. Human milk is part of a co-adapting triad of the mother, of the infant, including the milk, and being embedded in an environment that rapidly changes. So human milk changes, the triad changes and adapts to the environment. Now, if we wanted to reduce it just to human milk, you see on this graph here, which you're not supposed to see, at least not in detail, that there is so many different components of human milk, and each individual one has a function. And of course, in context and in concert together, they drive infant outcomes as well as maternal outcomes. So very complex topic that we need to reduce down to individual components to really understand what's going on. So let's just focus on the macronutrients of human milk and forget about all the micronutrients and all the other bioactives that we will talk about at the very end of this session. So in human milk, we have protein, fat, the milk sugar, lactose, and then we have oligosaccharides, 5 to 15 grams per liter, which often exceeds the amount of total protein. Now, if we compare that to bovine milk, there's about 100 to 1,000-fold less oligosaccharides. The structures are very different. And since bovine milk is the basis for most infant formula, we hardly have any of these human milk oligosaccharides in infant formula, although that is starting to change. Now, a quick word about the structure of human milk oligosaccharides. What does that actually mean? Oligosaccharides comes from the Greek oligosakkar, a few sugars, and that's exactly what it is. It is a few sugar molecules, in fact, 5 monosaccharide building blocks that are put together in different linkages, in different combinations to make an array of different human milk oligosaccharides. Those 5 building blocks you see down here on the bottom are glucose, galactose, N-acetylglucosamine, fucose, and sialic acid. And depending on how we put these building blocks together, we get different structures, different functions. In the blue spot up here, I'm going to show you what we call the HMO blueprint. So that's the general formula, if you want, of human milk oligosaccharides. And then the white space around it will give a few examples of some of those oligosaccharides that you may hear of throughout this talk and others. So all human milk oligosaccharides start with lactose at the reducing end, and then they get extended by disaccharide units in different linkages. They can be branched, they can be further extended, and that just alone is a repertoire of backbone structures that we call the neutral non-fucosylated oligosaccharides. Now, if there's non-fucosylated oligosaccharides, you can imagine there is fucosylated oligosaccharides, meaning we can add fucose to these backbone structures, and we can do that in different linkages. If we add fucose to just lactose alone, we get the small oligosaccharides, 2-fucosyl lactose and 3-fucosyl lactose. If we add it to the growing chain and in different linkages, we get more complex structures that we also call fucosylated. Now, these would be our neutral fucosylated oligosaccharides. Well, if there's neutral, there must be something opposite of that. So there is the acidic or silated oligosaccharides, and that's because we can add sialic acid to the growing structures. Again, in different linkages, we can start off adding sialic acid to lactose alone, and we get 3 and 6-sial lactose, and we can add it to the growing chain and just 1 sialic acid, as you can see down here at the bottom, and that would be our acidic or silated oligosaccharides. Now we can combine that and add fucose and sialic acid to the backbone, and that would be our acidic or silated fucosylated structures. So it's really a different way of putting these different monosaccharide building blocks together. Remarkably, there's about 150 to 200 different oligosaccharides that have been identified by structure so far. And really every mom makes a somewhat different fingerprint of what oligosaccharides are in her milk. You can see that here in data from the Canadian child cohort, where we see on the x-axis 1,200 different subjects, different milk samples, and on the y-axis the oligosaccharide concentrations in micromole per ml, different colors being different oligosaccharides. And it's evident how different that is between different moms. Low concentrations on the left, high concentrations on the right, the pattern being very different. And that is highlighted here again. If you take all those samples with a 100% abundance and now look at how the different oligosaccharides are distributed, it looks very different between different moms. So it's almost like a thumbprint. On the other hand, while we have huge inter-individual variation, we have very little intra-individual variation. So within the same subject, within the same mom, over a short period of time. So here you see milk oligosaccharide composition over the course of 7 days, and clearly there is hardly any difference. And if we go even higher in resolution and look at every hour, there is nearly a constant oligosaccharide concentrations here in the same subject. Again, very different between 2 different subjects. On the bottom now, a different subject, so the color pattern here looks very different for bottom and top, but looks remarkably constant over this short period of time. Over a longer period of time, you see that oligosaccharides, most oligosaccharides drop in concentration. So here we're talking not about several days, we're talking about from 1 month to 24 months, 2 years of age. And you see that many oligosaccharides drop in concentration with a few exceptions, one being 3-fucosyl lactose up here and 3-sial lactose down here. So there's a few oligosaccharides that go against the stream and increase over time, and we're trying to understand why that is. Now, here's a different representation of how oligosaccharide compositions can be remarkably different. This is data from about 10,000 milk samples, where every dot in this 3-dimensional space represents the HMO composition in a milk sample. And the closer those dots are together, the more similar the HMO composition, and the further apart, the more dissimilar. And you can clearly identify here that there is 2 different bubbles, if you want. So we call that HMO lactotypes, also known as secretors and non-secretors. And without going too much into detail what that means, it just means that there is different oligosaccharide compositions on the right than there is on the left. Both make oligosaccharides, but the oligosaccharide composition is very different. And these samples have been collected really from different parts of the world, but geography is not really what's driving this separation between secretors and non-secretors. Now, there is research focus on at least three different questions here. What are the maternal drivers that are responsible for the different HMO composition? Why is it different between different moms? And then, of course, what is the impact of these oligosaccharides on infant health and development, term and preterm? And last but not least, can we leverage some of that gained knowledge to develop new therapeutics for people of all ages, something we won't talk about in this presentation. Just one word or one slide really about the maternal drivers, since the focus should be on infant health and development for this presentation. We've seen that genetics, maternal genetics, are really the main drivers of HMO composition variation, to the point where this left and right distribution that I showed you earlier is driven by a single nucleotide polymorphism. So genetics play a major role in why we have different HMO composition profiles. But we've also seen lately that maternal nutrition, exercise, use of probiotics, maternal health, maternal drug use can all slightly impact human milk oligosaccharide composition. Why is that important to know? Because at one point, when we identify which oligosaccharides are good for certain infant or maternal health outcomes, we might be able to actually modify oligosaccharide composition by suggesting mom going on a different diet or using a different exercise regime or using a probiotic. So we're already setting the stage to potentially modulate oligosaccharide compositions if we find out which oligosaccharides are preferential. But now let's talk about infant health and development, which of course is the key interest. What do these oligosaccharides actually do? And what happens and how do we study that? So here's the research pipeline, as we call it, where we really use the entire spectrum from in silico models, in vitro and ex vivo cell models, and then go into animal models that are more or less suitable to study this. And then a very strong part is using human cohort studies, where we can analyze hundreds, thousands of milk samples for HMO composition, and then associate that with maternal drivers or infant outcomes. And last but not least, of course, we have clinical studies, and two of our speakers will specifically focus on that when it comes to HMOs and the current clinical studies. So all of that happens not only in one lab, it happens really in a network of collaborators from around the world. And since there's so many different topics where human milk oligosaccharides are currently under study, I want to focus on one specific aspect, and that is specifically on the very low birth weight infant, on the preterm infant in the NICU, and that is on necrotizing enterocolitis. So necrotizing enterocolitis is still a huge burden for the preterm infant. About 5% of all very low birth weight infants develop this often deadly disease. And on the other hand, we see that preterm infants who receive human milk are at a 6 to 10-fold lower risk to develop NAC. And we want to know what is in human milk that makes it so powerful to save preterm infants from this devastating disease. Now here in this space, I'm going to develop the long story of how we started to study human milk oligosaccharides in the context of NAC. So you'll see this slide come over and over and being filled in here, the white spaces. First, what we did is to address this research is to take an animal model, a rat model of necrotizing enterocolitis, and ask the question, if we feed these rats with pooled human milk oligosaccharides, so all of the oligosaccharides isolated from human milk, do we get any effect on necrotizing enterocolitis? And indeed we did. So if you look at the survival plots here, this is over the first 96 hours, you see that all the infants that stayed with the dam, all the pups that stayed with the dam, so the dam fed control here, all of the animals did not, none of the animals developed NAC. Whereas the formula fed without any oligosaccharides, we lost about 25% to NAC. Now, on the other hand, if we add human milk oligosaccharides to the same formula, you see that the survival goes up significantly and is almost to the level of the dam-fed control. So that to us was a huge positive surprise to see that oligosaccharides have such a positive effect here. We then went a little bit deeper looked at the intestines, looked at histology of ileum sections here. We're able to score that with the help of a pathologist. And not only is survival different, but also you see that the pathology scores from healthy 0 to disease 4 is remarkably and significantly different if we add human milk oligosaccharides to infant formula. Now we wanted to know, well, which oligosaccharide is it? So we started to use a multidimensional chromatography approach to separate the oligosaccharides in different fractions and subfractions. So we didn't have to test all 150, 200 different oligosaccharides in, let's say, 20 animals per oligosaccharide, which was, of course, completely unfeasible. So we first separate oligosaccharides by charge. And as I said earlier, sialic acid introduces a negative charge. So now you have acidic oligosaccharides, and you can see that here oligosaccharides separated by charge -1, -2, 3, and 4, and the oligosaccharides that carried a negative charge 2, so 2 sialic acids, were the most effective in reducing pathology scores. And we then looked at this. There's more than one oligosaccharide, of course, that has 2 negative charges, and in the second dimension we separated those now by size. So depending on how large the backbone is, we could separate that, and we then get a better resolution and find that there is one oligosaccharide here that is responsible for the protective effects that we see in this animal model. And we then used several different techniques here to identify how this oligosaccharide looks like, and we identified it as DSLNT, which stands for disialyl lacto-N tetraose. So it contains 2 sialic acids, disialyl, on this LNT backbone here. Now, that's great. That's an animal model. That is not a human preterm infant. And we, of course, now wanted to see, does this really translate to humans as well? So what we did next is we designed our own mother-infant pair cohort. Remember the power of cohort studies, where we specifically recruited moms and their preterm infants, collected milk samples that went into those infants about every second day, analyzed that for oligosaccharide composition, and then identified which oligosaccharides associate with a higher risk of NEC. And so briefly to go through this, you see that infants who develop NEC received milk with less of this protective oligosaccharide DSLNT than infants who do not develop NEC. The cohort was done in 5 different sites in North America. And here's an example how we matched cases and controls. Here on the top, you see a case. These are the first 28 days we had milk samples available every time you see a number in one of those boxes. These red boxes here highlights that the infant developed NEC during that time. So we had 4 samples before, 4 samples after NEC. And then we matched that based on gestational age, birth weight, delivery mode, when we have samples available, and so on, and then analyzed the oligosaccharides accordingly. So here's the results of that. If we looked at oligosaccharide total, so the sum of all oligosaccharides that we analyze over the first 28 days postpartum, we see there's no difference between controls, which is the dots, the gray dots here in the back, and the NEC cases, Bell stage 2 in yellow and Bell stage 3 in red. So there's no difference between controls and NEC cases for total oligosaccharides. But that wasn't the hypothesis to start with. The hypothesis was that there is this one oligosaccharide DSLNT that is potentially less in milk samples fed to infants that later develop NEC. And here is the data for DSLNT. It's exactly what we had hypothesized. Again, gray dots are the controls. Now the Bell stage 2 and 3 samples all cluster in the lower concentrations of DSLNT, which means there is less DSLNT, less of the protective oligosaccharide in the milk samples that was given to infants that later developed NEC. And here's a slightly different way to look at that. If we really look at each and every sample, color-coded now, so all these are our NEC cases. Red means the concentrations of DSLNT are lower than in the control, and all the samples here on the top for DSLNT show a red color, meaning DSLNT is lower than in the controls in every single sample. That's not the case for other oligosaccharides, so it's highly structure specific. And if we then model that and look at the odds ratios, you see that DSLNT really stands out as being significantly associated with a lower risk for NEC. So the more DSLNT, the lower risk for NEC, or the other way around. And we can then even go in and say, well, can we use that as threshold models where we say, well, if we take a milk sample and the concentration is higher than a certain threshold for DSLNT, then most of our infants will not develop NAC. If DSLNT levels are lower than that threshold, then we have to be careful, look at a few other parameters, and then make a decision whether we want to feed that milk to this infant or rather switch it or supplement. So this is great to screen mom's own milk, to say that your DSLNT levels are fine, you can give that to a preterm infant. We can also screen donor milk and human milk products like fortifiers to make a decision of whether we want to use that in the care of this patient. Now, these were 8 NEC cases in this study. We then collaborated with Dr. Chris Stewart, who's going to talk afterwards. And they have this wonderful biorepository of milk samples and many other samples from infants that develop NEC and have control samples for that. So we were able to analyze that. And here now you see we have 33 NEC cases, 37 controls. And there is clearly some difference here in the oligosaccharide composition. And if we narrow this down to which oligosaccharide is different, you can really see that there is one oligosaccharide standing out as being highly associated with NEC, and that is, again, DSLNT. So the more DSLNT, the less likely to have NEC. The less DSLNT, the more likely to have NEC. And Chris will talk about that in the next talk. So filling in the blanks of this cartoon here, so DSLNT not only was found to be protective in the animal model, it was also found to be associated with NEC risk. Less DSLNT, less of the protection, meaning higher risk for NEC. So that now gives us greater confidence to move into clinical studies, and we're going to talk about that in the future talks as well, and also to study the mechanisms of how this actually works. Now, to connect those two different things, we've shown that HMOs at least have some part in this protective mechanism of human milk again. And not only HMOs in general, but really DSLNT, one specific oligosaccharide in particular. We've seen that a specific HMO is associated with lower NEC risk, and the same HMO improves survival and NEC pathology in an animal model. This is, of course, a great opportunity to develop new therapeutics based on DSLNT. And it's also an opportunity to develop new diagnostics to screen mom's own milk and donor milk products. So this is NEC. This is one example for the preterm, very low birth weight infant. Oligosaccharides have been associated with many, many other things as well, mostly in the term infant space. For example, when it comes to diarrhea, pathogen exposure, and here's some examples for 2FL, Campylobacter, enteropathogenic E. coli, so potentially some protective mechanism. LNT has been associated with group B streptococcus infections, but also with protozoan parasites like Entamoeba histolytica. We've seen that other oligosaccharides are associated with rotavirus infections, and you can go on and go through different pathogen types, whether it's viruses, bacteria, protozoan parasites, and different oligosaccharides having potential protection. We also see there's oligosaccharides being studied in infant growth, and 2FL again pops up being associated with growth in infancy that extends to early childhood. Neo-LNT plays a role here because the ratio between those two is important. And we now have 3 or 4 different human milk cohorts or human cohorts where we show the same associations over and over and over again. So it gives us greater confidence that there is something real that's not just a bystander effect association. And then on the other side of the spectrum of growth and malnutrition, we find that the silated oligosaccharides seem to be effective here. One more example, neurodevelopment and cognition. We see that 2FL here again stands out being associated with memory and learning in animal models, but we've seen this recently in a human cohort as well, that there's associations between 2FL concentrations at 1 month and 24-month cognitive development in the infant. And then the silated oligosaccharides play a role here as well. And one more for immune development, different oligosaccharides being associated with certain cytokines that being released with chronic macrophage inflammation, which we study extensively. And there's a review that goes into this in detail and links different oligosaccharides with different immune responses. Very important here is to see that in the cohorts that we've done, we very often see that It's not an individual oligosaccharide that is associated with outcome when it comes to the immune response, but it's really a mix of different oligosaccharides, either being here associated with cow milk allergy, with food sensitization, or with allergic disease, short-term and long-term. And here we looked at all the way out to 18 years of age. So it seems to be of a mix of oligosaccharides being responsible. So a couple more words of how this all works. What is actually the mechanism? How do these oligosaccharides do that? And we strongly believe that oligosaccharides are more than just food for bugs. And I'll explain that in a second. But before we get to that, one word about what happens to the oligosaccharides actually when they get ingested. They're very resistant to the low pH in the stomach, also are not digested by pancreatic or brush border enzymes. 1% is absorbed, makes it into the systemic circulation, and is excreted intact with the urine. And then the rest is either degraded in the colon by bacteria or is excreted intact, and we find that in the diaper. And in this cartoon, you see really the interplay between oligosaccharides here on the top, good bacteria here in green, potential pathogens in purple, the intestinal epithelial cells on the bottom, and then underneath that, immune cells that are either associated with the intestine or act on a systemic level, since oligosaccharides can make it all the way through. So we see that oligosaccharides are prebiotics, so they are degraded, they're utilized by certain bacteria. Those bacteria have a growth advantage and potentially have a benefit on the host, on the baby. And all that can then feed back into the intestinal lumen here. And once again, there is local effects, but oligosaccharides make it into the systemic circulation, and there can be effects on organs, on tissues, other than those in the gut. So that's something to keep in mind. And last but not least, we clearly think that we are only at the very surface here, at the tip of the iceberg, and there's so much to discover what all these oligosaccharides can do. All right, so to summarize this introduction to oligosaccharides, HMOs are the third most abundant solid component of human milk. It's a group of complex sugars, complex glycans, and there's about 150 different ones. Some are small, like 2FL, 3FL, 3SL, and 6SL, or they're larger, like very strongly branched structures, high molecular weight structures. Then we have some that are either acidic, so they contain sialic acid, or they're neutral, no sialic acid. And then they can be fucosylated or not fucosylated. The structure determines HMO function, and this is important. Not all oligosaccharides do the same thing. There's a reason why we have 150 different ones. They're not digested by the host, by the baby. There's local effects in the gut, as well as systemic effects after absorption. They're serving as prebiotics, but they're doing so much more than just being food for bugs. They're anti-adhesive, antimicrobials, epithelial cell modulators, immune cell modulators, and a lot more. And that way they can affect outcomes like infectious diseases, diarrhea, growth, neurodevelopment and cognition, immune system development and response, and as I've shown you in the example here, particular necrotizing enterocolitis, but we also have some data on sepsis. And once again, this is probably just the very surface, and there's so much more to discover. Now, we see that HMOs contribute to shaping the infant gut microbiome, which may impact infant growth and development, and you will hear more about that from Dr. Stewart in the following talk. HMOs are prebiotics, but also more than just food for bugs. So really there is an opportunity to find the right balance of what oligosaccharides should be utilized when we develop prebiotics, probiotics, and symbiotics. Sometimes you really want to spare a certain oligosaccharide like DSLNT that is not utilized by your probiotic of choice. So it's still available to do other things. We need a combination of preclinical, cohort, and clinical studies to fully assess and validate the effective functions of HMOs. And Dr. Sayevska will show you in this session what kind of studies have already been done in the term infant. And then in the following, Dr. Hescott will talk about specifically the preterm infant. And last but not least, human milk is not just HMOs, right? I mean, it's my favorite topic, but I know there is so many other bioactives in human milk, and it's really a part of dynamic co-adapting mother milk infant triad, as we call it. And Dr. Cavallo will talk about that at the very end and give us a little bit of a future perspective of bioactives there are and how we tackle this really complex system of this triad. So I leave you with that and go to the next speaker, where we talk about more about the microbiome and what oligosaccharides can do there. Thank you. All right, so our second speaker of the session is Dr. Christopher Stewart. You saw some of his work already mentioned in my presentation. We started a great collaboration together a couple of years ago. Great guy to work with. So Dr. Christopher Stewart graduated in biotechnology from the University of Northumbria in the UK and did his PhD in molecular and microbial ecology at that same university. And before going to Newcastle, he worked at the Baylor College of Medicine here in Texas as a postdoctoral associate. So far has published more than 70 peer-reviewed papers, which is pretty amazing in this early stage of his career. Recently was awarded the Wellcome Trust Sir Henry Dale Fellowship to continue working on necrotizing enterocolitis, oligosaccharides, and microbiome, and has also patented a novel co-culture system for studying bacterial host interaction using an anaerobic organoid model, which is very powerful to really study these interactions between bacteria, host, and then throw some milk components in it. His current interests are in microbe host interactions in the preterm infant and health and disease in that context. But he's also looking at the other spectrum of the age here, looking at the role of the gut microbiome in response to biologic therapy for adult IBD patients. He will be talking about the importance of the gut microbiome in preterm infants. Welcome, Dr. Stewart.
Hello, everybody. Thank you very much to the organizers for the invitation. Thank you very much to Lars in particular. I've been a fan of this conference over the years. I've always watched the videos online after on YouTube, so it really is an honor to be able to present this year, although a shame not in person. The virtual format is still nice. And it's really nice to be following Lars as well in what looks like a really interesting symposium. I'll be picking up some of the concept I'm sure Lars has introduced around human milk oligosaccharides and building upon that with the importance of the microbiome in preterm infants. Some disclosures and disclaimers. And I think this audience particularly probably have a very good handle on what the microbiome is, quite literally the small community that occupies a distinct environment. For the purposes of today's talk, that will very much focus on the bacteria in the gut, but it's important just to be aware of other microorganisms that colonize the gut. So you have archaea and fungi, bacteriophage, that are viruses that infect bacterial cells, and also human viruses. And in adults, at least, around 56% of the cells in the human body come from our microbes, and less than half from our own human cells. When we consider that at the genetic level, less than 1% of all genes in the human body come from our human genes, and over 99% come from our microbes. And I think over the course of human history, we've had a very good understanding of the role of microbes in disease. And I think what we're increasingly appreciating is the role of microbes and bacteria in particular in human health. I won't go through all of these, but just to note a few that I think are particularly relevant for early life: resistance against pathogens or pathobionts. Providing that protection against epithelial injury is crucial for diseases like necrotizing enterocolitis, which I'll come on to talk about. And also something I won't talk about today, but I think is very important and will over the coming years, I think, be crucial in our understanding of complex diseases, is how the microbiome and the immune system interact during especially the first year of life, which is likely the key time frame for human development. And again, just to introduce the topic and what will be relevant when I'm talking about some of our recent preterm data, I wanted to introduce some of our previous work around term infants. And so here, this was a large cohort called TEDDY, around 12,500 stool samples sequenced from around 1,000 infants across 6 different geographical locations. And the study itself was focused on diabetes, but with such a rich data set, it could also be used to just define what factors are associated with microbiome development in early life. And that's exactly what I'm showing here. First of all, on the left, we're looking at the bacterial species. You can see our first time point of month 3 to month 6, and you see quite clearly breast milk is the main factor associated with microbiome development at that time period, and other factors, for instance, birth mode. If we fast forward to 1 year of life, again, you see breast milk is still the most important and most significant factor associated with microbiome development. Birth mode, we also still see, and other more environmental factors also significantly impacting microbiome development. After 1 year of life, we really no longer see much breast milk in the diet, and we no longer see that as being significantly associated with microbiome development. And indeed, by month 19 of life, each individual has become very individualized in terms of their microbiome, very stable, and is not really perturbed by any of the other covariates anymore. And that applies for the remainder of the sampling. So that was looking at bacterial species. When we look at bacterial function or functional capacity, we see that only breast milk was associated with the microbiome functional capacity, and again, only over the first year of life, after which breastfeeding really seldom occurred, and we no longer see that as being significantly associated. Again, this is published. I won't go into any further details on this paper other than this slide. And I guess behind the scenes of this slide is some quite complex modeling, which you can feel free to go and dive into the extended data within this manuscript. It's all open access. But just for visualization's sake, I think this is a really nice plot just to show breast milk declines from the diet over the first year and a half of life. You see those corresponding decreases in several of these different bifidobacterium, particularly Bifidobacterium breve. And this hopefully follows on nicely from what Lars was talking about in terms of human milk oligosaccharides. We know there's several functions for HMOs, and I'm sure there's many more to be discovered. But one of the primary roles is to serve as a prebiotic, particularly for Bifidobacterium. And so perhaps it's not a surprise here that as breast milk and HMOs are reduced in the diet, we see corresponding reduce or decline in some of these different Bifidobacterium species. There was further work done in a sister paper published by Tommy Vatanen, who's now in Auckland, New Zealand, looking at the HMO utilization genes of these different Bifidobacterium. Again, I won't present that data today, but it's worth having a look at if you're interested. And in essence, we observed that whilst breast milk was in the diet, there was much more abundance of genes responsible for the utilization of HMOs. And when breast milk was removed from the diet, those HMO utilizing genes generally diminished from the infant microbiome over time. So really that sets us up for our preterm work, which is now the primary focus of my lab here in Newcastle. I'm very lucky to work alongside clinical colleagues, particularly Dr. Janet Berrington and Professor Nick Embleton, who really have worked tremendously hard over the last 10 years to build a large research basis here in the northeast of England. And from infants born significantly premature, so less than 32 weeks at gestation, less than 1,500 grams at birth, they've collected a huge range of sample types, all of which I haven't listed here, but the main samples that I use for my research are described here. And you can see the various applications that we use these samples for. For instance, we collect maternal breast milk, and we, in collaboration with Lars Bode, profile HMOs. We also look at microbiome in breast milk as well as stool, and stool and urine we also profile functional small molecules by volatile organic compound analysis as well as metabolomic analysis. And in terms of more invasive samples such as salvaging blood and resected tissue, we have really good protocols in place that allow us to collect these samples that would otherwise be discarded. So not for research purposes primarily, but whatever is otherwise considered waste can come to us in the lab. We can use those samples to directly perform, for instance, microbiome analysis at the site of disease, immune profiling, and most recently to generate human intestinal derived organoids. And I'll mention a little bit about that work at the end of the talk today. All of these samples are housed in the Great Northern Neonatal Biobank here in Newcastle, some 100,000 samples from over 1,000 babies in that resource currently. It's available not just to myself locally, but also investigators around the world. So if you are interested, please reach out to us. We're always happy to help and support and collaborate with other research. And equally, we've got quite a lot of data now generated from these types of samples. If you're interested in accessing the data, very happy to share that. So here's an unpublished study, largely performed by my newest recruit, a PhD student, Lauren Beck. It's of healthy preterm infants only. All the data was generated using metagenomic shotgun sequencing. And here we have 124 preterm infants and nearly 1,500 stool samples. So a reasonably large cohort, certainly the largest healthy cohort with metagenome data to date. Much like the earlier study I mentioned around diabetes, we were very keen to define what covariates were associated with microbiome development in this exclusively premature population. And much to our surprise, factors like gestational age and birth weight were not significantly associated with the overall microbiome profiles. Delivery mode also wasn't, although we have published previously to show that actually we don't see delivery mode as being a main driver of microbiome in preterms, unlike, of course, in term infants where you do see that. This may well reflect the universal antibiotic use on the unit following birth. And I think so it's notable some of these absences, but also notable some of the associations that we did find. So antibiotic, breast milk, and breast milk fortifier all at some point were significantly associated with the microbiome profiles. And perhaps to nobody's surprise, probiotics were also significantly associated with an altered microbiome development at all time points except the first year. And this probably reflects that probiotics were used from day 8 on average. And they were stopped around day 44 on average, or on a median of day 44. So we do still see some effects afterwards, and I'll talk about that in the coming slides. Just to give you a little background onto some of the techniques that we use, which in and of themselves don't really tell us anything, but we can apply them to models and transition network plots that would otherwise not be possible to perform. These are dimensionality reduction techniques. So we use Dirichlet multinomial modeling, or DMM clustering. And this allows us to take all the 1,500 stool sample metagenome data that we have and distill it down into a defined number of groups. In this case, the algorithm determined 5 groups to be optimal. We then number those groups, 1 being the group that contains on average the youngest samples, and group 5 being that contains on average the oldest samples. You can see via this heat map the type of distribution of these different species that we have, largely confirmed here by our Lefse analyses. And really, just to give you a sort of very high level view of this, you see that Staphylococcus generally dominates cluster 1. Preterm gut community type 1 is what the PGCT stands for. Escherichia, both E. coli and unclassified, dominate preterm gut community type 2. Klebsiellas in number 3. Bifido-anomales and longum, particularly in cluster 4. And again, here you can see the Bifidobacterium breve dominate in these cluster 5. And again, that's reflected here in that heat map. So as I said, the real purpose of that is to allow us to do techniques and analyses that we would otherwise not be able to, the main one being this transition network type analysis. And so here's just a reminder of those dominant taxa across those different clusters 1 through to 5. And you can see here are different time windows. And this population in this particular view had no probiotics. So going back 10 years, we didn't use probiotics on the unit, and these were collected over the period of 2010 to 2013. And you see that most infants start in cluster 1, which is our Staphylococcus rich group, and that's quite common. And they generally transition towards clusters 2 and 3, so the Escherichia or the Klebsiella group, and they tend to remain quite stable within one of those 2 clusters once they transition there. In around 2013, middle of 2013, the unit introduced Influran probiotic, which contains B. bifidum and Lactobacillus acidophilus. And you see a very stark contrast to the microbiome transition in that population. So here, even within the first time point, you see some infants already have transitioned into preterm gut community type 5. Certainly throughout their sampling, they're much more likely to transition and remain within this preterm gut community type 5, which is our breve dominated cluster, which is interesting because breve is not part of Influran. And finally, after I think around 2017, we introduced Labinic or LB2 into the unit. And again, you see a very different profile from that to no probiotics, but also that to Influran influence. So although the probiotics are very similar, only difference being Labinic has an addition of Bifidobacterium longum subspecies infantis. You see that in those infants, they generally transition towards cluster 5, cluster 4, which is our B. longum, B. bifidum group. And it's worth noting, as I said earlier, we deliberately call these clusters 1 to 5 based on their average age as samples. So by proxy, clusters 4 and 5 could be considered our most mature. And so the two take-homes here are, I suppose, Infloran and Labinic certainly increased transition into our most mature gut microbiome community types, preterm gut community types, and also, regardless, they are both increasing the amount of bifidobacterium in these infant guts. Here you can again just see the very stark contrast between the two different probiotics. So our never group is in the middle, and you can see, depending on which probiotic they get, they sort of head off or drive in very different directions based on their microbiome profiles. So again, just underscoring the importance of probiotic selection, even when the same species are contained largely, clearly they're different strains and are having here very different impacts on the infant gut microbiome. So just to look at the prevalence, so less focused on relative abundance, but just whether or not we could detect those probiotic organisms. For B. bifidum, which was present in both, we were able to detect that before probiotics were given. So again, suggesting at least DNA from these probiotics is detectable in the stool before the infants ever received that probiotic. We detected during and after, which is less surprising, and much higher prevalence compared to our never group. You might wonder how we have a before, during, and after never population. That's simply just age matched based on the average ages of these different windows. And again, you see differences between the strains. So the Infloran B. bifidum has a much higher prevalence compared to Labinic, but of course, both are significantly higher than the never. Here we have B. longum, so that's only present in LB2 or Labinic. Again, we see the before colonization, as expected, again, during and after a very high prevalence. But interestingly, we also pick this up in Infloran as being significantly higher in our probiotic versus never group. And here again, lending support for this bifidogenic effect, where the presence of some bifidobacterium, in this case B. bifidum, is perhaps supporting the growth of other bifidobacterium, for instance, longum. And finally, Lactobacillus acidophilus, which was in both probiotics, is higher than the never group at all these time points. Again, we're detecting it before. And I think it's always striking for Lactobacillus acidophilus, especially how sort of rare it naturally occurs in preterm infant populations, yet is used quite extensively throughout different probiotics. Okay, so now I'll talk briefly about some of our published work. And again, a disease that probably needs no introduction to this audience is necrotizing enterocolitis, the leading cause of death in preterm infants, and is basically an exaggerated inflammatory response leading to necrosis of the bowel, most commonly the terminal ileum. And in the worst cases, those infants will undergo surgery to have those regions resected. So we know that human milk oligosaccharides and the microbiome have been associated with this disease through our work, through Lars's work, and many others around the world. And so this has been an area of great interest for us over the last decade or so. Just to pick up on an animation from a paper published a few years ago now, here we have 6 preterm gut community types because we're dealing with 16S data rather than metagenomic data. Don't ask me why it clusters 6 ways with 16S and 5 ways with the other, but it does, and that's pretty consistent in our hands. Here, each dot represents a different stool sample, and it's colored based on which cluster that infant sample derives. So basically, the yellow here is our bifidobacterium dominant cluster. You'll see during the animation, our neck infant and control infant both start in the Staphylococcus rich group. The neck infant shows much more chaotic or less stable development over the initial weeks of life, contrasting that with our control infant who moves gradually from the Staphylococcus rich group into the bifidobacterium rich group and then remains in there for the remainder of their sampling. So this is 2 arbitrary cases, but I think it makes the point quite well. We applied statistics to this in the published manuscript, again, open access, and showed that infants who went on to develop NEC had significantly less stability and significantly less relative abundance of bifidobacterium. So we followed this up in collaboration with Lars to look at maternal human milk oligosaccharide profiles, showing that NEC and control infants have significantly different HMO profiles, or infants who go on to develop NEC versus those who remain healthy controls have received significantly different HMO profiles from mother's milk. Very nicely, and this very rarely happens in science, when we looked further into see what HMO was driving this difference, it was the same HMO that Lars had shown in his earlier work, being diacylylacto N-tetrose, was really driving that difference between NEC and control. So a really rare moment in science where you get really good corroboration of previous work from a completely different area, different numbers in a different population. We applied some univariate models to that, determining 241 nanomoles per mil would be the optimal threshold in our cohort. This would require validation and more cohorts to begin to make that more generalizable. But within our NICU, at least, we would have very good sensitivity and specificity using a threshold like this. And you can see here basically how our control infants generally almost always above that, and our NEC infants generally below that. And finally, using that clustering approach, we also were able to show that infants who had above the DSLNT threshold generally progressed into those clusters 4 and 5, which is our more mature microbiome profiles dominated by bifido, and infants below that threshold generally did not transition towards clusters 4 and 5 and remained in the more staph and other pathobiont-rich clusters. Okay, I promised I would briefly mention the organoid work that we have ongoing. For those unfamiliar, when we get tissue samples from our preterms, we isolate the crypts, generate these 3D enteroids. We then differentiate them into all the major cell types of the gut. And we have this model whereby we can keep the bacteria in anaerobic conditions, the human cells in oxygenated conditions, and then model how they interact over a period of time. And we've shown very nicely using preterm versus adult enteroids or organoids that there's a clear difference. And what I thought was really interesting here is when we add the microbiome, that's when we see the biggest change in expression and gene expression between preterm and adult derived organoids. So lending some evidence towards this being a useful model for studying premature disease such as NEC. I won't go through the summary. If you're watching this as a recording, you can press pause at this point. I would like to thank the various members who's been involved in this work, particularly Andrea and Lauren, to Janet and Nick, without whom none of this would be possible, and to Lars for his support with the human milk oligosaccharides, to our funders, particularly the charities and with whom we work very closely. And with that, here's any contact information, and I would be delighted to answer any questions you might have. Thank you for your attention.